So good Friday morning, and welcome to the third episode of the second season of the Backyard Naturalist series, Raiders of the Lost Archaea. Whether you're with me live on Friday morning or watching this recorded, I consider it a privilege to be spending this time with you. The Backyard Naturalist series is brought to you by the UEC In My Backyard, which features content for people of all ages. I thought we had the last submission from communication intern Elizabeth Gamillo a few weeks ago, but even though her internship ended a month ago, we still are getting a couple final submissions, and she put together this video on how to make a backyard birdseed bell. So the next time you feel like scrolling through your social media, maybe consider scrolling through this website instead. It's a lot better for your soul, in my opinion. Also, this is super exciting news. I mentioned last week, I mentioned that the Yardversity project surpassed the 1,000th observation. And in this past week, we reached the milestone of our 500th species. And we asked some of the folks tuning in live here to think of their best guesses as to what the backyard species was that was number 500. And we are going to reveal it now. It was, I'm sure you were all thinking of this, a black bear, because depending on where you live, and this is a reminder that you can contribute to Yardversity from anywhere in the world, and for some of you, black bear is a backyard species. So it's a surprise to me and a pretty cool species to have at number 500. If you're interested in participating in Yardversity, please contact myself or Ethan or Maggie and join us for one of our monthly Yardversity events, which is soon to include trivia for adults in the evenings. So we're looking forward to that. And then finally, I'm excited that Hike Milwaukee starts Sunday and runs through the week. It's an annual fundraiser for the Urban Ecology Center that we've had to adapt to current conditions. You can sign up for solo guided hikes, complete with an accompanying podcast, or there are small group hikes led by staff or volunteers in groups of 10 or less with masks and distances. For more information, please visit the Urban Ecology Center website. And I'm a little late to register this year, so if you don't want to sign up for a hike, you can contribute to my fundraising page. I'll post it on Facebook later today, or shoot me an email. But I encourage you to go on one of the hikes if you can. And with that, we'll move on to the featured group of organisms. Now, just in case you misheard the title of this talk, it is not about the Swedish furniture conglomerate or a park in Maine, but it's pronounced slightly different. But IKEA actually does provide us with a good introduction to Archaea, because what happens when you open up an IKEA box and then you open up all the other boxes that are in the boxes, and then you find some bags and in the boxes with lots of little screws and dowels and all this basic stuff that, you know, by itself isn't that exciting. Or maybe show somebody Legos for the first time. You know, it's cool. It's a bunch of colorful blocks. And at first blush, that might be what you're thinking about when you think of Archaea. I mean, you know, what's there to get excited about? You can't see them without intense magnification. They don't migrate millions of miles or add beautiful color to your backyard. But then when you take a closer look at what you can kind of make with each of these three sets of ordinary materials, these IKEA pieces produce a beautiful Brimness bookshelf. These Legos, out of these Legos, you can make models of Archaea. And although it's not a direct relationship, or it's not as direct a relationship, Archaea likely represent the most primitive form of life on Earth, which eventually led to things like this peacock mantis shrimp. So Archaea are kind of can be thought of as building blocks, or the raw material for just about everything that followed in life, we think. When we look at the division of life, then we'll start with Archaea, because it's very likely that the first organisms on Earth most closely resembled Archaea. You'd have to go back a little further to start with some way of forming and then replicating organic molecules, leading to something similar to RNA, which then later began to code for cells, but then after that, you have this very basic structure of a cell with a membrane and the genetic material and some ribosomes floating around in a cytoplasm. The second major division or domain is bacteria, which, like Archaea, are considered prokaryotes, but differ from Archaea in some really exciting ways, like how they make their cell walls and membranes, and how they replicate, and how they manufacture proteins. But please don't fall asleep yet. I promise it gets better. In fact, getting better happens right now with the third domain of life, which are the eukaryotes, which include things like this. And the major difference between eukaryotes and prokaryotes has to do with the cell again. So we have to say goodbye to our otter, or do we? So once again, Archaea and bacteria are considered prokaryotes, which translates to before nucleus, meaning all of the DNA in the cells is just floating around willy-nilly because there are no internal organelles to organize things. The eukaryotes differ in that our cells, and I'm using the term "our" because we are all eukaryotes, our cells have internal structures like a nucleus, which provides this tidy storage space for DNA. And how eukaryotes evolved from prokaryotes is material for another lecture, but if you've ever played Pac-Man, consider the eukaryotic cell as Pac-Man, and the nucleus and ribosomes and all those other organelles are like the dots. And it's not too far off to say that eukaryotic cells evolved when cells started swallowing other types of cells to form what's called this endosymbiotic relationship. And so the swallowed dots started working together with the Pac-Man in this mutually beneficial relationship to form a more advanced cell, which I'll call Miss Pac-Man. And then the next big step was then these advanced eukaryotic single-celled organisms started collaborating with each other to form colonies of advanced cells, which then led to colonies of advanced cells in which cells on one end specialized in eating and cells on the other end specialized in pooping. And then you have these advanced multicellular organisms. So there's a very brief story of how Archaea became us. And because Tori's here, and as she knows, you really can't finish a lecture on anything, including Archaea, without at least one good poop story. So the word Archaea, and I've seen it spelled both with that extra A and without, means ancient or original in Greek, which is fitting for what is likely the oldest form of life. But one of the really crazy things is that, as Jenny mentioned, we didn't even know that Archaea existed until my lifetime. So they weren't discovered until 1977, even though they are literally everywhere. And because they looked a whole lot like bacteria, we classified them as bacteria, and we gave them the name Archaebacteria. And then we moved our familiar bacteria into a group called Eubacteria to distinguish them. And at the time, most organisms were classified by similarities in function and behavior. If they looked alike, if they behaved alike, we grouped them as closely related. But even after their discovery, we really didn't learn much about them because they don't grow on petri dishes. And so they're really hard to cultivate and study in a lab. And when you find out later where they can grow, this doesn't make any sense. But this was also about the time that researchers started looking at and classifying organisms by their genetic material rather than the structures and behaviors. And then it wasn't until 13 years later, in 1990, when we were in IB biology, that we realized Archaea are not a form of bacteria, but if we did go back to our biology texts, we would likely still find this relic. So in 1990, the three-domain system was proposed, and we realized that Archaebacteria are not bacteria, and so we gave them their own domain called the Archaea, and then Eubacteria just went back to being plain old bacteria. They're about the same size as each other, and they function similarly. Eukaryotic cells are usually much bigger, but the Archaean bacteria, the cells are about the same size, and they function similarly. So it's easy to see why they were grouped together for so long. And they both live everywhere you can possibly imagine. So whether it creeps you out or whether it makes you feel less lonely, there are millions of bacteria and archaea living in and on you right now. And so these next few slides, I'm dedicating to all those bacteria and archaea that are watching from your skin. And we'll take a closer look to decide which group is the toughest. So bacteria and archaea are the two main groups that make up what we call extremophiles. And these are organisms that live in extreme environments where they test the limits of things like temperature and salinity and radiation and lack of moisture. And in most cases, they can only live in these extreme environments and nowhere else. So if you gave a drop of water to an archaea that was living in extreme salt conditions, you'd be sending that individual into a death spiral. So again, we talked about what our ideal environment is. And for me, the ideal environment that I'm going to admit publicly is either sitting in a tub of water at about 97 degrees or so, maybe a little more, or relaxing in air temperature of about 72 degrees, as long as it's in the shade. So I'm not going to mention anything about crisp autumn walks. But I imagine that most of you would agree with me. Maybe you prefer slightly different temperatures, or maybe you're wrong, but I imagine most humans are good in these conditions. But for many extremophiles, these conditions would literally kill them. And they would quickly seek out water that's, 200 degrees or full of heavy metals or so acidic that it would burn our skin. So we'll take a quick tour of some of the places where extremophiles thrive. In the episode from season 1 on petrichor, or the smell of rain, mentioned a cyanobacteria that had been frozen in tundra permafrost for more than 500,000 years and remained viable so that when they were unfrozen, they continued to function. So these are examples of cryophiles or cold living organisms. Lake Untersee in Antarctica is not only cold, but it's full of methane and is about as alkaline as laundry detergent, with a pH over 11, which would kill a human very quickly. But there are both archaea and bacteria that thrive there. They'd be considered alkylophilic and psychrophilic organisms. Or places like Yellowstone with lakes that reach extremes in heat and acidity and sulfur. Not only do extremophile archaea and bacteria thrive here, it's actually where archaea were discovered, and different species are different colors based on the temperatures of the water in which they thrive. So that's why you see that gradation from hotter to cooler, and the bacteria and archaea that can thrive there. So these would be considered thermophilic and acidophilic. The mud deposits of Mona Lake in California are devoid of oxygen, highly alkaline, extremely salty, and full of sulfides. Yet there's a party of bacteria and archaea, along with brine shrimp, and there's even a species of fly that lives in the mud that can create their own air bubbles. But if you moved most of these organisms up into the atmosphere, the oxygen would kill them. Yet before we had oxygen in the atmosphere, the environment was more like what they're used to. So they were likely the ones that were thriving before plants and other things changed our atmosphere into what we need. Iron Mountain in Northern California has water that's so full of heavy metals and acids from decades of mining that if you left a shovel in the water in the morning, by nightfall, it would become what we call a handle. And yet even in these highly, highly polluted waters, these scrappy bacteria and archaea are going about their business. If you wanted to open a spa business for archaea and bacteria, you could just pop the top off a nuclear reactor, because there are extremophiles that thrive in heavily irradiated nuclear waste. Or you could just put an open sign on a rock, as some extremophiles can live in tiny, tiny, tiny fissures in rocks. So upon this initial review, we're going to call it a tie so far, because for all of these extreme environments, we found both bacteria and archaea. But we're starting to learn more about archaea, and if we search a little farther, or more specifically deeper, we find that archaea take this extreme living a little farther than the bacteria do. So deep on the ocean floor, kilometers below the surface, we find these underwater volcanoes called black smokers that are hydrothermal vents in which the seawater becomes superheated by magma below and erupts just like a geyser. There's plenty of life that can survive near these vents, even with this extreme pressure. And bacteria can get close to the openings of the vents, surviving in water that's just under boiling. But as we all know, you can get rid of bacteria, you can kill bacteria by boiling the water. And so archaea wins that dare contest about who can get closer. They can get right up into these smokers in water that's actually above the boiling point. It doesn't boil because of the pressure, but they can withstand water that's 221 degrees Fahrenheit at least. So far, that's what we know. So archaea win in that environment, and they also win in extreme salty environments, salt deposits. So some salt pans can reach 99% salinity, which is just almost pure salt, and that kills even the hardiest of bacteria. Yet we still find these archaea hanging out like it's no big deal. So archaea so far are the winners in who can survive more extreme environments. But as I mentioned earlier, for whatever reason, we have a really hard time cultivating archaea in a lab like we can with bacteria, because archaea can survive just about anywhere on Earth, but not in petri dishes. Go figure. So there's still a lot that we don't know about archaea because we're just now learning how to grow them in the lab. Although I think of how much fun it would be to try to recreate the heat and pressure of a black smoker volcano in a lab. But because of this quirk, there are still many unanswered basic questions, like how do they live and how do they interact with other organisms? A major reason to celebrate archaea, if you go away with one thing, is that unlike bacteria, archaea are not harmful to humans. There's many bacteria-borne illnesses, but there's no archaea-borne illnesses that we know of. So that's pretty cool, and maybe a good way to start the friendship with all the archaea living in you. But we are starting to find out ways to study archaea in the lab, which are leading to some very cool potential applications for science and engineering. And this all started with the discovery of the first species of archaea, Thermus aquaticus, in the hot springs of Yellowstone. And it produces a polymerase enzyme, which is the basis for industrial applications of PCRs, which allows scientists to replicate DNA billions of times in just a few hours. So it's a big process that led to huge breakthroughs in genetics throughout, since its discovery. So when you hear in forensic science, when you have DNA being used in an evidence, that's due to this discovery from archaea. If you go to any of those genealogy programs, like Ancestry.com or 23andMe, when you swab your cheek and send in those cells, or you swab your dog's cheek, or however you do it with dogs, you have archaea to thank, or curse when those results come back. And it's kind of cool to think about it this way, that the technique that archaea made available then also allowed the further study of archaea itself, which led to further genetic breakthroughs that now, as of like the past few years, are now finally allowing us to start studying archaea in a lab. So another major application of archaea in industry comes in the form of wastewater treatment and biogas reactors. So one of the extreme environments that archaea thrive in is our stomachs and our guts. And many archaea are methanogens, producing methane in your gut. So the next time you let out a particularly smelly toot, Tori, don't blame the dog. You can just blame the archaea in your stomach. And in a wastewater treatment plant, Archaea produce methane for biogas reactors, which produce energy for humans. So the more we know about the optical conditions for archaea in these wastewater treatment plants, the more energy we can derive from them.
Hey, Tim, we have a question going back to the water temperature in
Yep.
thermophiles in the Yellowstone.
Yep.
And they're wondering, does that mean that the species that turn blue in the center would turn yellow or red in different locations?
Mm.
Kind of like hydrangeas are pink or blue depending on soil pH.
I don't know anything about hydrangeas, but that's an excellent question. And I'm going to hold off answering it because I'm going to kind of get into that process later. So a good question and a good reminder about just how crazy this stuff is. I thought you were going to interrupt me about the smelly fart, so I'm glad we're getting away from that immature gibberish. So, but we're even using Archaea as applications to improve laundry and dishwashing, which may sound like a joke, but your washing machine and your dishwasher actually mimic these really hot alkaline environments that are found in these extreme natural environments in which Archaea can thrive. So a lot of applications there. They also produce enzymes used to study the treatment of autoimmune disease. They're used in tanning leather and removing hair from hides. They're used in making paper. And again, we're really just scratching the surface because we're just starting to figure out how to study Archaea. And so I expect there's going to be some pretty big advances in the coming decades now that we're finding out how crazy they are. But probably my favorite application for Archaea has to do with extraterrestrial life. And several extremophiles, including one species of animals, this bizarre and wonderful microscopic tardigrade, which can survive weeks in space despite the complete lack of oxygen and intense solar radiation. And it makes sense that we learn how to deal with space, which is an extreme environment, by studying the critters that can live in extreme environments on Earth. So extremophiles, particularly Archaea, are leading to some practical implications for space life, like how to improve spacesuits and other aspects of an astronaut's life. But Archaea also likely provide clues to the potential for extraterrestrial life. So you may have heard recently a study that showed that life may exist in the upper atmosphere of Venus. And there are many speculations that conditions could allow for life on Mars or some of the moons and the planets in our solar system. And if we were to find extraterrestrial life, chances are pretty high that they would resemble Archaea. And some scientists speculate that Archaea-like creatures may have evolved first on Mars and then later reached Earth through a process called panspermia, interplanet travel. And if that were the case, then we are all the descendants of aliens. But on the flip side, we do need to be careful when we send rockets or people to other moons or planets, because we could unknowingly seed life that just might thrive there. And you might think that that's too unlikely, but with all these recent developments, we learned that Archaea cells actually share the same proteins that human cells have related to epigenetics. And so there was a recent lab study, now that we can cultivate them in the lab, that looked at the most extreme Archaea for salt tolerance. And then the researchers decided to crank up the salt level to 11, higher than any natural environment we know of, and thinking it would likely kill the Archaea. But they were actually able to evolve quickly to that extreme salt environment. But evolve isn't the right word. It was through epigenetics. So the DNA sequences of the organisms didn't change as they adapted to that higher salinity environment, like ultra high, like testing the limits, higher than anything on Earth. And uh, there were no, I should say the DNA sequences didn't change from before and after. So the new environment caused structural changes in the DNA that caused for new genes to be turned on and off. So that's, again, this idea that you can change in your lifetime. Evolution doesn't have to happen generationally. And poor Lamarck, who was ridiculed for his thoughts of the giraffe stretching their neck, making it longer, that some of those aspects were actually based in sound science. So they're expecting some pretty big advances in the study of epigenetics, partly because you can study them in archaea, and they might be the ideal study organism for that. And this gets to your question about the Yellowstone and the color changes. We've known for quite a while that bacteria are pretty good at exchanging genetic information, and they do it through a variety of processes, including viruses. But this is, for example, how antibiotic resistance spreads among bacteria. And so we know that they can quickly exchange genetic material. But what we've learned from archaea takes this concept a step further. Archaea also have many different ways in which genetic information and genes can spread. But scientists have found cases where two completely separate species, and this doesn't happen with bacteria, two separate species will get together, exchange genes, and form a new species on the spot. And as we learn more about these, we're starting to better understand how sexual reproduction began, or how eukaryotic cells evolved. And if you go back to my Pac-Man analogy, we're now pretty sure that the Pac-Man in the relationship was archaea, and the organelles that it ate were a bacteria. And if you watch the episode on mycorrhizae, I kind of left with an image of this forest in which the trees were communicating with each other, sharing resources, even if they weren't part of the same species, warning each other of danger, this kind of like this community, this idyllic community of socialism. Well, even though it's not, they're not aware of this, but it turns out that archaea are likely behaving in very similar ways. We now think that archaea form these networks of molecules where communication happens, and as a group and as individuals, they're able to make split-second decisions with each other. And they're likely, in a sense, warning each other of stresses, just like trees do. And stress likely induces the quick exchange of genetic material. So someone explained it as if let's say you're one of those salt-loving halophilic archaea in the lab, and then someone puts a drop of fresh water in your environment that will likely kill you. That stress will trigger a massive exchange of DNA, with the hope that, probably a lot of the combinations won't survive, but a few combinations will arise that will be able to survive that stress. So it's like they're saying, "Oh no, fresh water. Hey, Ethan, quick, give me some of your genes to see if it'll help." And so this kind of gets to your question too about the Yellowstone. If you were able to take organisms from one color and move it into the other, it's probably likely that some would die, but they may be so used to those changes in temperature and pH that it may be that since they've been evolving with them so much, the epigenetics are there to change an individual within a lifetime. Or it may be just that this quick evolution happens so fast that it could be changed within a lifetime, or it could be changes within generations. So it's pretty fascinating and mind-blowing when you think about how that works and how really advanced these single-celled organisms are when you look at them as a network, just like when you look at a colony of cells working together. So it's a little bit of a shorter episode because there wasn't a whole lot I can find, but I now think I have a new favorite domain of organisms. And I'm looking forward to all the crazy new things that we're going to learn from them. So if you're in school now and you're looking for a career, study Archaea. Seriously, you can come up with breakthroughs like the recent study that tells us you have more Archaea on your skin under the age of 12 and over the age of 65. So we're going to end with a brief video because sometimes I think when you hear all this information, explanations of these topics are better done through this form of media. But I'd like to thank you all for joining me this morning. And after the video ends, I'll stop sharing my screen, and you're welcome to stick around for a few minutes if you have questions or thoughts or your own stories related to your backyards. Here we go. We'll give this a shot. It's a cute little video.
Have you ever imagined things with a movie trailer voice? You know, that dramatic voice that commands you to listen and be intrigued that shows up on some movie trailers? We think it could actually work great for biology. An example? In a world with no oxygen, one little yeast cell will do fermentation. Okay, tell me that does not make you want to learn at least a little about fermentation. If so, we have a video about that, and the yeast, which is a fungus, which can do it. But you know what would be the best topic for an exciting biology movie trailer? Archaea. Because they are probably some of the most exciting unicellular organisms out there, and many of them can live in extreme environments too. By extreme, I mean think harsh, but then more harsh. Areas with extreme temperatures or high salt levels, though I should point out Archaea are not the only extremophiles. Bacteria can be extremophiles too. It's interesting because when I first started teaching, it was common to call Archaea Archaebacteria. But that is an outdated term because Archaea aren't bacteria. Now, they do have some things in common with bacteria. They're both unicellular, they're both prokaryotes, so they have no nucleus, and they also don't have membrane-bound organelles. They both have genetic material and cytoplasm and ribosomes. Both bacteria and Archaea can be classified as autotrophs or heterotrophs, and they both do binary fission to reproduce. Still, bacteria and Archaea are two completely different domains in the three-domain system. That's a pretty big separation. What makes them different? In the three-domain system, you have the domain bacteria, the domain Archaea, and then the domain Eukarya, which has all the eukaryotes: plants, animals, fungi, protists. So let's look at the difference between bacteria and Archaea. Membrane structure is very different. Remember that all cells have a membrane. It's really important for homeostasis. If you take a look at our further reading suggestions, you can explore how the chemistry of the cell membrane of the domain Archaea is different. One example is that Archaea membranes typically contain ether linkages instead of ester linkages. You would find ester linkages in the membranes of bacteria and Eukarya. Some Archaea have a lipid monolayer instead of a lipid bilayer. Turns out that a monolayer, as well as some other structural differences of Archaea membranes, can be useful for surviving in those extreme environments. You will also find a unique cell wall in Archaea. Recall that you will find cell walls in bacteria, and you'll also find them in Archaea too. Archaea cell walls can be very diverse, but they don't have this large polymer called peptidoglycan. You will find peptidoglycan in the cell walls of both gram-positive and gram-negative bacteria, while Archaea can also technically be gram-positive or gram-negative, most Archaea staining gram-negative. Archaea don't have the peptidoglycan. Archaea can have pseudo-peptidoglycan though, which is a little confusing, but it has a different chemistry to it. Like all organisms, Archaea have genetic material. Like bacteria, Archaea DNA is generally arranged in a circle. Although while bacteria tend to have one origin of replication, Archaea tend to have several. When studying the genetics of Archaea, it's interesting that now scientists find more similarities between the genetics of Eukarya and Archaea than they do Archaea and bacteria. So overall, these were just some comparisons of Archaea with bacteria. Keep in mind that most Archaea can't be cultured in the lab. That's because there is a lot unknown about what kind of media they need to grow on, or nutrients or environmental controls in order to grow them in the lab. Although, that's a common challenge for quite a few types of bacteria too. Before we go, I want to mention my very favorite Archaea genus. So I have not been to a volcanic spring yet. Need to travel more. But Archaea members of the genus Sulfolobus have. They can handle temperatures that most organisms can't, like around 80 degrees Celsius, hot. And that's not all. This genus can also handle very acidic conditions. Substances such as hydrogen sulfide, which is dangerous to most living organisms, do not deter them. There's interest in studying how these archaea could be used to treat industrial waste, and even convert parts of that industrial waste into something that could be useful. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
And that's it.